using wearable devices. This is a really passion of mine. Let's start with looking into what glaucoma is. Glaucoma really is a devastating disease. It's the #1 reason for irreversible blindness in the world. In US, nearly 1% of the population, 4 million people, is diagnosed with glaucoma, and there is no cure. So it's progressive nerve damage. I mean, in that sense, it's similar to Alzheimer's. It's a neurodegenerative disease. The reasons are not known. There is no cure. The main risk factor for glaucoma is the intraocular pressure, the pressure inside the eye. And that's the only modifiable risk factor. There are other risk factors such as age, genetics, etc. But the intraocular pressure, the pressure inside the eye, is the only modifiable risk factor. What do I mean by that? We can actually prescribe drugs according to the intraocular pressure of the patient, and we can try to decrease the pressure of the patient. Okay, so that's the goal. If the pressure is too high, that can cause the more severe, more quick progression of the disease, so the patient becomes blind even sooner. So that's okay. I mean, we can measure the IOP, we can measure the intraocular pressure, but the problem is, one interesting feature of the intraocular pressure is it changes during the day. So it just varies. Okay, so here is during the 24-hour period, the IOP goes up and down. Okay, so there's a large variation, as you can see. So that's a problem. And how do we measure intraocular pressure? So you go to the doctor's office, doctor basically touches on your eye with a probe. Okay, so it's similar to, I mean, this technique, I'm not going to go into the details, but it's similar to getting a basketball on your hand and just poking it, right? Just pushing it. If it's really hard, you know that there's a lot of pressure inside. If it's really soft, to the thumb, you know that it's not that high of a pressure. So very similar, okay, very similar. It works really well, except that it is uncomfortable. And you have to go to the doctor's office. Doctor tells you what the pressure inside the eye. Okay, so he has to, or she has to tell you what the pressure inside the eye. You cannot do it by yourself. So one-time measurement is not really ideal, right? I mean, we understand that the intraocular pressure changes during the day. So one-time measurement is not enough to manage the glaucoma. So how can you solve this problem? So here is one technique that has been suggested, using a contact lens wearable device. So if we have a sensor, we call the sensor a strain gauge. Okay, strain gauge. You can see it here. This is a contact lens the patient puts into the eye. There is a strain gauge. Strain gauge actually changes shape as the pressure increases inside the eye. The cornea shape changes, and this cornea shape change causes the strain gauge to change shape. Okay, so that's a smart way. Okay, and the way we measure this, the way this strain gauge works is basically as the shape changes, the resistance of the strain gauge, the resistance, electrical resistance of the sensor changes. So if you look here, the resistance is a function of length and area, as you can see. So if the length changes, the resistance will change, right? So we see that. Let's not focus on the antenna and microchip, but you can see that they are bulky, they are metallic structures, so we don't really want these type of components in a contact lens. It can be uncomfortable as well. And you might be thinking, I mean, what the antenna is doing. So antenna needs to talk to an external receiver. So there is an external antenna around the patient, as you can see, and this needs to be there also. Okay, so it's not only the contact lens, and there needs to be an antenna around it, around the eye. So there needs to be this data recorder that needs to be used. So overall, I mean, it makes the price of the components a little high. Nobody wants to have a taped antenna around their eye, so that's not ideal as well, okay? So we want to have some alternative to using this metallic strain gauges, electrical measurements to the strain measurement technique. So how can we do it? So let's look at what's out there. First of all, what is a strain gauge? I mentioned it already, but I want you to know how a strain gauge works. So I said that the resistance, electrical resistance, is a function of the geometry, but also the material properties. So this resistivity rho here that you see, Greek letter rho, is a material property, it's a resistivity property. Typically, strain sensors look like that. So it works based on the change in the length and the area. So and we can tell that the length and the area affects the resistance. We talked about it already. And these type of devices have limited sensitivity. There is another approach which we call piezo-resistive sensors. These piezo-resistive sensors have some nanoparticles inside. Okay, so we dope the material with nanoparticles. So now, because as you apply the strain, as you stretch a material, the nanoparticle separation will change, the distance between the particles will change, so this resistivity will change as well. It's a fancy word, piezoresistivity, but it's simply the material itself changes its characteristics, changes the resistivity, so it has very high sensitivity. So that's another approach. Okay, so but the problem is with that is it's a little bit difficult to achieve directionality, and it's a little bit costly. Okay, you need nanoparticles, etc. So what was our question to begin with? Can we use microfluidic variables? Okay, so microfluidic variables. What is a microfluidic variable? So this is basically what's out there, and an alternative to both of these geometrical strain sensors and piezoresistive strain sensors is a microfluidic strain sensor. So it's simply a thin polymer film. It's a soft, stretchable film with some microfluidic channels. So maybe this is more suitable for contact lens applications. Okay, let's think about it, and maybe we can try to I can try to convince you, or you will think something else. So because there is no metals, there is no microchip, there is no antenna, it is a little bit more comfortable compared to the other strain sensors that we've seen. But you might be asking, how is it going to work really? So this is basically a schematic of what we're looking here. So this is the exact same thing. Okay, so it has a channel. It's what we call a liquid reservoir channel, this blue one. There is an air reservoir, and there is a sensing channel, which is the red one. Okay, so you might, I mean, this is a little bit complicated right now, I know, but it is really not so different than a standard mercury thermometer. You might have seen this at your homes. It's simply a liquid reservoir and a connected sensing channel. This one measures temperature. This one on the left senses strain. Okay, so it's very similar. So how do they work? The liquid level inside the sensing channel, as you can see, changes. So it tells you, this level tells you what the temperature is in a temperature sensor, and this level will tell you the strain level in a microfluidic strain sensor. Okay, so that's the same principle. So now let's try to have a look at how this, this idea works a little bit closer maybe. So the volume inside the sensing channel changes as we apply the strain, and volume is a function of length and area. So it's similar to the resistance in that sense, but it's a little different. There is no material characteristics, so there is no dependence on the material resistivity. Okay, so this is exactly the same structure that I showed you. It's just we're looking at it from the side, a little bit simplified. So the liquid level inside the sensing channel tells us what the strain is. When we apply the strain, the liquid level changes. So this is very similar. I mean, you might be having a hard time imagining this. So let's have a look at this. This is the same reason when we're walking on the beach, when we step on the sand, the wet sand gets dry, right? This was the first GIF that I showed you at the beginning of my presentation. So when you have sand particles, when you apply force to it, you create a volume increase, which creates a vacuum and pulls the liquid inside. So this is what we're seeing here. So you can imagine that you can measure the force that is applied to this balloon by looking at the level of the liquid here. So same principle. So the volume change, okay, will be proportional to the liquid displacement and inversely proportional to the basically this cross-sectional area of this neck, as you can see. Okay, so the narrower the cross-sectional area, there will be more liquid movement, liquid displacement. So it's the same principle. So, but how a channel volume changes when it's strained. So it's a little bit difficult. I'm not going to go into the details at this short presentation, but it's basically when you have a piece of rubber band, when you stretch it, it's going to get longer, but it's going to get narrower. If you have a microfluidic channel in the middle of this rubber band, it's going to collapse a little bit. Okay, so you can see that it's collapsing a little bit, but still volume will increase, believe me. So here you see that when we apply the strain, this structure is elongated a little bit, but the membrane is collapsed a little bit. I mean, typically in a contact lens, you can imagine that we want to make this membrane thinner and thinner. So when you make the membrane thinner, there will be more collapse, but there's a solution to this problem. You can make the membrane thin, but you can have a very narrow structure, which increases the stiffness. So now it's very stiff. It doesn't collapse as much. So, okay, so we now have a way to have a lot of volume change in a microfluidic channel. We can make them narrow and we can put a lot of channels side by side. So concentric rings. So there will be a huge volume change when we apply a very small strain to this device. And then we can put it inside a contact lens and we can test it on animals, which we are doing in our labs. I mean, of course, this is a porcine eye, not live animals. This is inoculated eyes. And when we compare it with the results from a tonometer, similar to what I showed you by poking the eye. So we got for example, very nice results comparable to this tonometer devices. So this is a combination of, again, my research, my several classes. So this is the end of my presentation. I think I'm at the end of my time limit. So if you have any questions, I can take one or two questions. Then if not, I'll leave the stage to Dr. Mobed-Miremadi. Okay, so thank you, everyone. So I'll leave the stage to Dr. Mobed-Miremadi.
Hello, everyone. I'm trying to get the video going. Thank you, Dr. Rossi. I don't know how I can top what you showed. I'm going to open another presentation, and I will be brief. To share screen. Okay. I've done so many presentations, and yet just one moment, please. I have problems with my screen. think my computer is freezing. One moment, please. My sincere apologies. My computer is freezing. I'm requesting 2 more minutes of your time. Computer. About 25 years ago, I started working in the area of artificial cells. And this is basically the area of bioseparation and biomembrane. And what I do here at Santa Clara University, I'm a senior lecturer. I teach courses in the area of biomaterials, biostatistics, and bioenergy. So following up on what Dr. Rossi was talking about, the field of biomaterial and device design encompasses all these subfields right here. And he did show you how he had designed a device from scratch to go from a prototype all the way to the preclinical or translational realm. So I will be focusing today on artificial cells that go back almost 50 years, but they're still used because there is a societal need for them. So some applications of artificial cells are they're used in regenerative medicine for cell encapsulation. They're used in water detoxification. They're used in charcoal detoxification, packed bed columns. And we have 2 types of artificial cells if you classify them by size. The first size is the submicron nanometer size particles that we inject for drug delivery and cancer therapy, targeted therapy. The second type are the implantables, the ones that are used in packed beds or artificial organs. And an example of a biomaterial that has been used for a very long time is alginate. Alginate is extracted from algae, and it comes to us in a powder form. And over a course of 30 years, it has been proven that by manufacturing alginate and implanting it can be a temporary solution until patients receive organs. This is an example of a study that was done. These are called artificial cells. These circles that you see here They're in the 50 to hundreds of microns. And what they do is they contain mammalian cells, and these mammalian cells are encapsulated within this capsule. So they cannot be attacked by the immune system. And this membrane is semi-permeable so that nutrients and oxygen could go in and out, and the cells can continue their function. So for example, if you are suffering from diabetes, and I know that there are many types of the progression of disease of diabetes is different for everyone. They have tried it encapsulating these implantable, these islet cells in capsules, and they can remain implanted for 6 months without being attacked by the immune system and regulate a patient's blood sugar. So going forward, for example, such a solution can be translated into a cartridge technology, a cartridge that could be changed daily that could regulate blood sugar. So the societal motivations, of course, are all these heart diseases, kidney disease, diabetes. And of course, there are competing technologies. We all hear of CRISPR, TALENs, Zingerfin nucleases, 3D printing, ghost organs. But in the interim, until these solutions are validated, these artificial cells can serve as test beds for multiple therapies, for testing regulatory approaches, and also they're a perfect tool for education. So the capsules that we use we try to stick to water sustainable chemistry. We do not want to generate extra solvent. So for example, one of the materials that we use is chitosan. Chitosan comes from crab shell. And in order for this material to be used, it needs to be characterized by calorimetry and by many other tests before it can be water soluble, before it could be absorbed on a biocompatible surface, and eventually be ready for injection. We do have an animal center in the new STEM building here. And this is an example of research work that was not done in university, but as a result of this formulation, we managed to extend the half-life of some implanted materials in mice. So this is why I have the translational aspects. So we talked about alginate. This is a structure of alginate coming from algae. This is membrane right here. This is a cross-link membrane that you see here. And this is a scanning electron microscopy of the membrane right here. And the way we make these capsules, or the way we make these structures, is using different methods of bioprinting. This is something that we teach here. And these bioprinted materials, they can be in a circular form. They can be in the form of a stent. And they have different mechanical and diffusive properties that enable us to accommodate solutions towards therapies of different diseases. So this is an example of microcapsules. These are larger, these are smaller. And for example, these are tailored for dialysis or lowering the urea concentration. Another area where microencapsulation is useful is in the area of storage and cryopreservation. It has been proven that cryopreservation of cells and it re-enables their regeneration. And right now, cryopreservation technologies are much needed because we have therapeutic materials that are produced and stored over a long course. And what I've taken the liberty to do is the name of the undergraduate authors from that participate in this work have been highlighted in green. So we have a very active faculty student publication program. This is another example of artificial cells that were extruded through this microneedle device, this custom SCU patented microneedle device. And these are the undergraduate authors that were co-authors of the patent, and they participated in this research. Currently, microneedles are used for a variety of applications, including vaccine delivery. This is a hydrogel bioassay, a biosensor. These are the undergraduate authors that have worked towards papers. This is also a simulation of degradation of what the biosensor is doing. We also have materials and related to biomaterials and related to BioAI. If you have a membrane, it means that your membrane has to be selective. Your membrane needs to have a pore size. So we are doing some intelligent pore size classifications using atomic force microscopy. The pores that you see here are in the nanometer range. And we're predicting whether a membrane can be used for a therapeutic purpose or not, and to which extent. The material of this membrane is cellulose or cross-linked cellulose. And cellulose comes, of course, from trees. It's the second most abundant biopolymer. And we are also extending the use of these biomaterials to biofuels. And we're trying to extend the sustainability applications of these biofuels. So these are examples of including these artificial cells that are made from these biomaterials in these bioreactors, and they're immobilized for bioethanol production towards clean energy. This is an these are example of undergraduate authors here. This is a microbial fuel cell towards energy generation. We are using urea to fuel a fuel cell. And this is an example of trying to reduce the carbon dioxide in the atmosphere using a photobioreactor using biomaterials that we use in this program. So with my apologies for the delay, I'm ready. If you have any questions, I would like to answer them. Does anybody have any questions? Yes, no? All right. So then I think we're done. I don't see any questions in the chat.
I think we do have one question. Someone's asking,
Yes,
does
yes.
Kendall De Philippi work with you?
Yes, Kendall De Filippi works with me. She is been with me since last year. She's been conducting independent studies and research. And let me tell you specifically what she's working on right now. She's working in the area of this microneedle research. And her research consists of pre-screening the health of microencapsulated cells that are used in the antibody production against COVID. So this is an example of a project that she's working on with her partner, Dwight Johnson. So their research will impact the cost effectiveness of vaccine production.
I do have a question. So what would you say is the advantage of the location of SU in the Silicon Valley in terms of, engineering and everything you've just went over?
I think the location is openness, especially I think to start with, quite a few of our faculty have had, they're either in the summer, they do research with industry, or they come from industry. So they have outside connections that validate or invalidate the usefulness of effort and design. So designs have two elements. They have an academic element that is invaluable. You cannot put a price on, but there is also many societal problems that need to be attended to. Perhaps you may not come up with a perfect solution, but you may come up with a solution in the interim that may enable to solve societal problems. So I think in the Silicon Valley, we get very quick feedback of whether at least short term are the problems that we're solving and the project that we're assigning to the students have any societal impact. I hope I answered your question.
So we have another question, and they're asking, how often do SU graduates go to graduate school with big names like Stanford, Berkeley, Caltech, MIT, etc.?
Actually, we do have a spreadsheet with that has a track record, and I would like to go revise it before I answer that. I would like to take your contact information. The last I looked about, and these are maybe the statistics that I'm telling you, they may be a year behind, but about 30% to 40% of our graduates go to graduate school, and about 5 to 10% end up maybe in those R1 institutions. I would need to revise those statistics. And when I say end up in R1 institutions, I mean they may end up there for their PhD program, or they may end up there for their law program. They may end up there for medical school, dental school. But I would love to have an email so I can, or a contact so I can provide exact and revised statistics, because we do track all those statistics. We do keep track of those statistics.
I sent my email in the chat.
You said my email in the chat? Okay. Okay. Make sure. I'm taking a picture of it. There you go. Hold on 1 second, so I don't lose it.
Okay, are there any last questions before we go ahead and wrap it up? No, okay. All right, thank you so much, Dr. Mobed-Miremadi. This was a very interesting session. I don't know anything about engineering, but I did learn a lot. If there's anything that you would like to say before we end the session.
There's something I want to say, that we have a very, very diverse group of students in engineering, and especially in bioengineering, about half or more than half are girls. So if you're thinking of giving it a try, I think you should really consider giving it a try. I think once you come in the program, this illusion around STEM, that girls can do it and things like that, it just goes away. There's a rhetoric, and then there is action, and I can tell you there's action in the School of Engineering for girls, for diversity, and a lot of other things. And I think that the best thing is, maybe you can contact us. I can put you in touch with some of our students, because I can say whatever I want to say, but I think peers make a big difference.
That is very true. Thank you for that, Doctor.
Thank you.
If you would like to provide your contact information so our students can reach out to you if they have any questions after the session,
Sure.
that would be awesome. But thank you all so much for coming. Thank you for joining us. And I hope this session was useful for you in deciding whether SCU is, the place you would like to study engineering at. So thank you all to both our professors that attended and presented. And I'm going to go ahead and let you all go. We do have our next session at 6 p.m. So you do have time to rest in between, but thank you all for your time.
Thank you. And I just want to say Dr. Rossi's email is also on the website. So every professor's contact is on the website. So, and we are very approachable. So, all right. Thank you. And I